This set of experiments that we're doing really builds upon work done a number of years ago by some of my colleagues, not only in academia, but also here at NASA, where it was demonstrated very nicely that colloidal particles, which compose soft matter, in fact, can be used as model atoms. Okay, atoms are hard to see, and colloids, which are relatively large, say on the order of a micron or so, actually you can see under a microscope and do those experiments where one can tune the interactions between them and see the results. We, in these experiments that Professor Wu and I are working on, are trying to build on that foundation in a couple of ways. One is to extend from colloidal atoms to molecules. Okay, so we're looking at systems which are not only single particles, but in fact are chains of particles with the associated dynamics and interesting behaviors associated with their structure, things which we hope will mimic biology, for example, so DNA or polymers. That's sort of one direction. And the other is the idea that we can use fields to control their behavior. So turning what was a passive system into something which is active. We have been working specifically on applications which are biomedical in nature. Imagine, for example, I could take these colloidal chains, or similarly, we take individual particles and form wheels with them so that they can move in the body, and with that, deliver drug or provide mechanical action in order to alleviate stroke or other diseases. The building blocks of the molecules, colloidal molecules that we're making, are on the order of 1 to 5 microns. Okay, so something on the order of 50th to a 20th of a size of a human hair. The advantage there is they're easy to inject because they're small. And once inside the body, we then assemble into larger bots, which can transport to the site of injury or where we want to target our delivery. We're also currently working on the idea of delivery into the lungs. The specific advantage here is that the individual particles, this colloidal material, colloidal particle, are now small enough that we can aerosolize them for inhalation. So you get them small enough, they go into the lungs, where we can then subsequently assemble into bots which are large enough, strong enough, powerful enough to potentially burrow through mucus. You know, this is certainly important in the context of COVID and other diseases where one often typically dies from just issues with respiration. The ability to get oxygen through those mucus layers within the lungs is really where the problem lies. And if we can come up with, microrobotic technologies that can help alleviate these issues, I think that would be a significant benefit. In the context of active matter and active materials, we've assembled individual particles, these colloidal building blocks, into lots of different things and controlled them. And that does include microscale pumps and microscale mixers and microscale valves. So these components would work together in order to make small factories which function together in a highly parallel fashion. You can imagine doing a blood test on a single drop of blood, something like that, or you could do 1,000 blood tests on a single drop of blood. That's really one of the holy grails of lab on a chip, and doing it all in a very, very small condensed environment certainly saves space, allows one to push to handheld devices and things like that, which would be very convenient, out of the doctor's office, both on the ground and in space. The systems that we're looking at, these actively controlled chains, as well as actively controlled wheels, are mostly plastic, but inside contain magnetic domains, iron oxides that allow us to control them within the field. Now, these materials actually are heavier than water, and as a result, it's very difficult to study their dynamics on the ground. Okay, they'll just sink. One can think of approaches to try to alleviate that. However, as we move into more complicated geometries, more complicated systems that are composed of multiple components, there's no way to use strategies on the ground to alleviate the settling, the issues that would basically mask the dynamics that we want to study. The space station obviously provides us a platform where we can study their dynamics free of interaction with gravity, free of interaction with the surrounding capillaries, glass surfaces, things like that, in order to get a pure reading of their dynamics and the structures that form in space. We would need a technique that works in 3 dimensions, absolutely, and something which would integrate external field control with 3D imaging is going to be critical. The systems that we're looking at are under the broad category of soft matter. They are colloidal molecules, ones which aren't simple atoms that we can image in a 2D plane. We need to see and characterize their behaviors in fully 3 dimensions, which requires confocal microscopy. Think of it as a baseline, right? We need to know sort of the passive behavior before we can truly use external fields to design the active behaviors that we're seeking. So having experimental observation to guide us in building theories that allow us to better model behaviors is going to help us develop new materials and better active matter when we start building microrobots and other things that are hopefully will perform a lot of the functions that I've been talking about.